All right, so, ladies and gentlemen, welcome to Bio 180. And today, we are going to be starting a brand new module. Hopefully, you just finished your first exam covering general chemistry. And so the next module is going to be focused on a topic that we're going to loosely call organic chemistry. Okay? And organic chemistry is the study of carbon-containing compounds and molecules. And so the reason why we call this unit organic chemistry, we're going to start out talking about carbon and how carbon is used to make different chemical molecules, biological molecules. From there, we're going to talk about other elements, nitrogen, hydrogen, and oxygen, that are used to enrich the carbon skeletons that we'll talk about today. Those are called functional groups. And then our last four lectures are going to focus on four categories of biological molecules. So we'll talk about carbohydrates, we'll talk about lipids as a group of compounds, we'll talk about proteins and nucleic acids. But today, we're just going to focus a little bit on carbon and mention a little bit why it's significant. And then we're going to work on learning some terms associated with isomers and spend a lot of time with isomers. And we'll describe why we're doing that in a couple of minutes. So if we come back here and talk about carbon. Carbon, the main statement is carbon is the basis or the basic element of life. Of course, there's other elements needed for life, but there's a reason why we often refer to life on Earth as carbon-based life. Okay? And so what makes carbon so special? Why is carbon the primary element of life? Well, the biggest reason has to do with its valence. So carbon, you learned in earlier lessons, is tetravalent. So carbon is tetravalent, which means that carbon has 4 valence electrons, and therefore also has a valence of 4 openings that it can form bonds with other elements. So by tetravalent, really what we mean is that carbon has the ability to form 4 bonds. Okay, now the other elements, oxygen will form 2 bonds, nitrogen will form 3 bonds, hydrogen will form 1 bond. So carbon represents the largest number in that group. There are other elements that form more bonds. So phosphorus, we'll see, forms up to 5 bonds. Sulfur can form up to 6. But carbon, with its ability to form 4 bonds, and in addition, a couple other properties that make it unique is carbon is lightweight, and carbon is abundant. Now, these last two might not seem very important, but if you consider, for example, other elements that make 4 or more bonds, silicon lies right below carbon on the periodic chart. And the reason why this lightweight becomes important is if you look at carbon, so carbon can form 4 bonds with oxygen and form a molecule called carbon dioxide. The normal state of carbon dioxide is a gas, meaning that carbon dioxide, and it's in the air, it's plentiful, and it's readily available because it's a gas and it's spread out all over. Silicon dioxide also makes 4 bonds with oxygen. And so silicon dioxide, which is just one element below carbon on the periodic chart, because of its heavier weight, is actually a solid. And so it's one of the main components in sand. And so even though there's a lot of it, there's a lot of sand on Earth, it's not readily available. It's not found in all environments, and you can't uptake it very well. So the fact that carbon is lightweight, its oxidized form, carbon dioxide is a gas, its fully reduced form, nothing but hydrogen surrounding it, is a molecule called methane, also a gas that can spread out and be readily available, biologically speaking. So these elements combine to make carbon sort of the main element that we study for life. And that's why we have a whole branch of chemistry, organic chemistry, that's dedicated to its study. Now, the rest of our time together, so that's carbon and why we study it. The interesting thing about carbon is it can make, because of those 4 bonds, it can make a variety of structures. Makes a variety of structures. It makes a good scaffolding by which we can start to build biological molecules. And so, starting with just carbon, we often refer to these as skeletons. So you can have carbon skeletons. So carbon skeletons. And this means the basic framework. When we talk about carbon skeleton, we're talking about the basic framework of carbon atoms. And they can have a lot of variations. So some ways that they can vary in what we call the chain length, or the number of carbons. They can also vary in what we call branching. So, what does branching mean? Branching is where you can have, for example, 2. So that's one carbon skeleton. It's just 4 carbons in a row. And I can draw another carbon skeleton. Again, it also has 4 carbons. So these 2 molecules that I've drawn up here would have the same chain length, or we'd say the same number of carbons. But notice they don't look the same. This one has 3 carbons in a chain, and then it has, this would be an offshoot off of that main chain, and that's what we would have referred to as a branch. So this would be 2 variations. They have the same carbon number, but branching causes a different variation to be formed. Okay? Another way that we can start to vary is by the formation of double or triple bonds. So we can have multiple bonds. Again, we can take this same 4-carbon chain, and we could rewrite this. Now we have a new variation of that 4-carbon chain. What's the difference? Still has 4 carbons. There's no branching. The carbons are all lined up in a row. But now we've included a double bond right in the middle of that chain. So that's another variation. These 2 would be 2 totally different molecules, different properties. Another way that they can vary is in what we call ring formation. How would that vary? We can take our same 4 carbons, and instead of putting them in a line, I could put them in a ring. I could take one end, bend it around, and hook it up or bond it up to the other end, and I would get a molecule that would look like that, and that would be ring formation. Again, just using 4 carbons, you can see that I can have a variety of structures. You start adding more carbons, 5 carbons, 6 carbons, and then I can get even more forms of these different variations. Okay? So just using carbon, because of its 4 bonds, we can get a wide variety of different molecules. There's one more. Notice that these 4 only dealt with changes in just carbon, the skeleton itself. We were talking about how carbon makes 4 bonds. Hopefully one of the things you noticed is that in these structures, I haven't been actually drawing all 4 bonds onto the molecules. I've just been playing around with carbons and connecting them. Where a final source of diversity comes from in the carbon framework is, notice I have all these positions that were unfilled, and I can start to attach what we refer to as functional groups. Different atoms or collections, even groups of atoms, onto those, and that can also create a wide degree of diversity. So I could take my same 4 carbons, and the most common group that we see attached to it is just a simple hydrogen. And so I can attach hydrogens to make up my 4 bonds. But then I can also attach other groups or atoms. And we'll learn a lot more about this in our next lecture. But here at the end, I've attached a different group. That's called a hydroxyl group. And so this molecule, if this one was nothing but hydrogens, and this one now has a hydroxyl group, I just created a totally different molecule. Okay? And so we can get quite a bit of variation in carbon structure using these 5 strategies. Okay? Now, what we want to do is, as you go through the course, there are different sessions. We talk about biochemistry, or when we talk about the different macromolecules, we'll talk about amino acids, we'll talk about carbohydrates and monosaccharides, like glucose. And you're going to see a lot of chemical structures. And so it becomes important in the course to be able to read and actually to some degree write or draw chemical structures. It also becomes important to really be able to compare and contrast those chemical structures. You might be given 2 amino acids and asked to identify which amino acid is which, or look at 2 structures and identify the functional groups that are present. So we have this needed skill of being able to read and write chemical structures and also be able to compare and contrast chemical structures. And then we also want to be able, just in general in the course, to give you some opportunities to practice and exercise analytical thinking. And so, how are we going to accomplish all of those? We're going to spend the rest of our time talking about isomers, okay? And there is some need, as you go through biology, there are terms like we'll talk about geometric isomers, we'll talk about enantiomers, and these do come up with biology. You do need to understand them. You probably don't need to understand them as much as we're going to talk about them today, but that's because understanding enantiomers and geometric isomers is not our primary purpose in going through isomers. Building these skills, and you as students, is our primary purpose. Okay? And so as we're going through, that's what we're trying to do, is give you a set of rules, allow you to apply those rules, practice analytical thinking, and be able to read and write some chemical structures. Okay? So just keep that in mind as you're like, I know a common question is, why do I have to learn this? When am I ever going to use this? We're teaching you it, if you learn it, if you decide to learn it, in order to develop these skill sets that do become valuable in the course and actually later on as you go through your careers. Now, as our central focus, how are we going to learn about them? We have one task that we want you to learn how to do. And that is, given a pair of molecules, like I've drawn up here on the board, can you identify the relationship between these 2 molecules? Molecules, like people, have relationships to each other. In molecules, it's based upon their structure and how similar their structure is. In people, we often base those relationships on their DNA and how similar their DNA are. For example, some human relationships you might have are parent and offspring. That's a relationship. So if I put 2 people in front of you, and you say, oh, that's the daughter of the mother, and there would be a relationship between them. You might take 2 other people whose DNA was more distantly related and say, oh, those 2 people are cousins. They have a relationship, and cousins is the word that explains that. The words we use for chemicals to tell whether they're related or not are isomers. Okay? And so isomers is like cousins or family relationships in people. It describes the relationship between 2 molecules. And that's what we're going to go through and trying to define some of these words today, and then we'll apply them to solve this problem. And we'll practice that a couple times. So, given any 2 molecules, let's come back over here to this side of the board. given any pair of molecules, we could then state that they are going to either be non-isomers or they will be isomers. This means they'll either be related to each other or they won't be related to each other. So what is the basis for judgment? What makes something an isomer? Something is an isomer if they have, the question we ask is, do they have the same molecular formula? Okay? If they have the same molecular formula, then they're isomers. They're related. If they don't have the same molecular formula, then they're non-isomers. And it's really that simple. Now, sometimes you'll be given that molecular formula and then it would be really easy. For example, they might say C6H12O6. That would be the molecular formula of one, and then you could look at the other one, C6H12O6, and you say, oh, those two molecules have the same molecular formula. They're isomers of some type. If I said, well, one of them is C5H10O6, then you could look at these two and say, oh, they have different numbers of carbon, they have different numbers of hydrogen, so those two are not isomers. They have a different molecular formula. It gets a little bit harder when you're faced with a pair of structures like this, because there's no molecular formula. So what do you do in this instance? Well, you count. And there's a certain order. It's probably easiest to count, much as I've written up there. The first thing I would look at is the number of carbons. So I'd say this one has 1, 2, 3, 4, 5 carbons. I go over here, 1, 2, 3, 4, 5 carbons. And so you could write that down here at the bottom, C5 and C5. The next thing we'll look at, we could look at hydrogen. That's what's in the formula over there. On the other hand, I tend not to, because there's probably more hydrogens than anything else on there. And so that's a lot more counting, and I don't like to count. So the first thing I'm going to look at after carbon is going to be oxygens. Because oxygen it's easy to identify, and it's a little bit more rare than hydrogen is. So I'm going to go down and kind of go around the molecule. So I've got 1, 2, 3 oxygens. So here I'm O3. And then here I've got 1, 2, 3. So here I'm also 3 oxygens. The next one, we always go through the big four first. So that's carbon, oxygen, nitrogen would be the next one. How many nitrogens do I have? Nothing, 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 nothing. 1. And 1 here. So I would write nitrogen. Now I could count my hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So that's H11. How many do I have here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Those are the big four. I might look now, are there any other elements represented on either molecule that I've missed? In this case, I've gotten them all. Be careful, look for things like a Cl, a chloride, or a chlorine molecule that might be put on there, fluorine, phosphorus. You might find some unusual elements. Those will usually be present in just 1 or 2, okay? There'll be 1 or 2 atoms maybe if they're present at all. So, the question I pose to you, are these then isomers? Well, they have the same molecular formula, so yes, they're isomers. Now, the next question we ask then, so we can go up here, let's erase these. If they are isomers, so let's see, they will, all isomers will pair out into one of 2 groups. So if they are isomers, they will either be what we call a structural isomer. So this would be yes, they are the same molecular formula, or they will be what we call a stereoisomer. So in our decision tree, they're the same molecular formula, yes, then it's one of these two. And if our answer is no, then we would have non-isomers. They would not be related. Now, let's talk then about the definition of stereoisomers and structural isomers. Structural isomers, what do we ask? Do they have the same bond pattern? Okay? Now, what do we mean by bond pattern? Let's go back. Earlier, we drew up these two molecules, these two carbon structures. So we had a straight carbon chain, and we had a same number of carbons. So we'll say that these two have the same molecular formula. But notice if we were to number these carbons, so let's put 1, 2, 3, 4, 1, 2, 3, and 4. The bonding pattern isn't the same. Up here in this carbon, we have a 1 to 2 bond, we have a 2 to 3 bond, and we have a 3 to 4 bond, right? Down here, we have a 1 to 2 bond, we have a 2 to 3 bond, and then this one is actually a 2 to 4 bond. And it doesn't really, you could do these A, B, C, D, you could renumber them, but you'll never end up with the same combination in those two patterns. To do this, to make this molecule right here, which is called isobutene, we had to break this bond and reform it on the middle bond. And so that's what makes then these structural isomers. So if the answer, do we have the same bond pattern, and that answer is no, then we have structural isomers. Okay? Now, if we ask that question and we say yes, they do have the same isomers, then we are looking at some form of what we call stereoisomers. So then we could say that structural isomers do not have the same bond pattern. Stereoisomers do have the same bond pattern. Okay? Over here, we can come over to our molecules over here, and we can start to ask that same question. To ask it, we're going to kind of go through each molecule and see if it has the same things bonded to it. So when we look at this first carbon up here, on this side, I've got a double bond to oxygen, I've got a single bond to a hydrogen, and then I've got a single bond to kind of the rest of this big chain. So I look over here. Now, we don't care how it's bound to that same carbon. We just care that it's bound. So for right now, you might be noticing, oh, this double bond to oxygen seems to be right here on my right hand, and over here it's on the left hand. We don't care about that for now. All we care about is this carbon has a double bond to oxygen, just like this one did, a single bond to hydrogen, and then a bond to the rest of the molecule. So it looks like that has the same bond pattern as that one. And then you go through and you look at the rest of these carbons, comparing them, and you see if they have the same bond pattern. So right here we would see carbon is bound to an OH group, an H. These group we already said was the same, and then the rest of the molecule. So those two are the same. Does that make sense to everybody? Now, what would it look like if they didn't, if this were structural isomers? How could I change and be a structural isomer? Well, if I remove that H and this NH2 and swap them, H and H2, Now, these two before would have been stereoisomers. But now, in the blue, because I swapped those two groups, they no longer have the same bonding pattern. This third carbon here has a nitrogen on it. The third carbon here does not have a nitrogen on it. This carbon has a nitrogen, and then its companion does not have a nitrogen. So these two then would be structural isomers. Okay, they do not have the same bonding pattern. All right, I'm going to change these back. And so far we've learned how to identify the difference between a non-isomer and isomer. And then if it's an isomer, we learned how to tell the difference between it being a structural isomer or a stereoisomer. If it's a structural isomer, we're done. There's no more form of relatedness. Okay, we're just done. We don't have to go anywhere, and we would just write structural isomer. However, we determined that those were actually stereoisomers. Stereoisomers can be divided up into three different categories. And so we have geometric isomers, we have diastereomers, and we have enantiomers. Now, how do these differ? Stereoisomers differ one from another by what we call a chiral center or a chiral carbon. And to understand a chiral carbon, let's come over here. And talk for a moment about chiral carbons. And then we'll apply what we learned to determine geometric isomers, enantiomers, and diastereomers. Chiral carbons come in two different flavors. So one form of chiral carbon is double bond carbons. What's a double bond carbon look like? It's a carbon that is participating in a double bond with another carbon, and that becomes significant. So it has to be a carbon-carbon double bond for it to be a chiral carbon. Now, when we draw it like this, the double bonded carbons actually start to form a structure. They form what's called sp2 hybrid orbitals. And so here's two. Remember, each carbon has 4 bonds. Here's 2 of the bonds in the double bond. And then the other 2 bonds kind of spread out in an X structure. So you kind of make a separated X structure where these 2 spread out as one half of the X and these 2 are sort of spread out in the other half of the X. And so that would be right here. Then each one of those carbons is called a chiral carbon. And the reason why it's called a chiral carbon is because those carbons we can't spin or rotate. So as an example of this, earlier today I made this little molecular formula or structure, ball and stick model. And you can see on the side that it has a double bond in it. These 2 springs represent that double bond. These are single bonds. And notice that then the black atom on this side and the yellow atom, I can't spin those. I can't put the 2 yellow atoms on the same side because if I let go, it rotates right back up. The double bond will not rotate. That means that whatever orientation, black, yellow, yellow, black, this was formed in, the double bonds were formed in, that is the orientation it stays in. It can't move out of that orientation. It's fixed in place. Okay? And so, anytime you have one of these carbon-carbon double bonds, then you get the atoms attached to it fixed in one of two positions. And this becomes important to know and identify. So we can either have, so we'll assume that this is for us always going to have a carbon and a hydrogen attached. There'll be some kind of carbon chain. And then we'll be having two hydrogens. So this is one pattern you might see. So here's my double bond, kind of splitting the board this way. And in this case, notice that I have a carbon on the top, and then the other carbon is also on the top. So the two carbons are on the same side. This is referred to as a cis orientation because it's cis is Greek prefix. It means same, on the same. And so the two carbons are on the same side, and so they're cis. The other orientation that you can have is what I've already drawn up here. And this orientation, now we have the two carbons on opposite sides. This carbon is on top, this carbon is on the bottom of the double bond. If we were to draw a line through that double bond, one carbon's on the top, one carbon's on the bottom. And this is called the trans conformation, or trans double bond. So one form of chiral carbon is double bond carbons, and because that bond cannot rotate, the atoms that are attached to the carbons become fixed in space, and so you have two possible orientations. You have a cis orientation, or you can have a trans orientation. Okay? This then becomes the basis for what we call geometric isomers. So geometric isomers, if they're going to be a geometric isomer, must have double bond. So we'll put a carbon-carbon double bond. So it must have a carbon-carbon double bond. And they must be in opposite orientation. Meaning that in order to have a pair, remember we're always talking about a pair of molecules, in order for that pair to be geometric isomers, they have to have a double bond, and that double bond has to be in opposite orientations. One has to be cis, and the other has to be trans. If they're cis and cis, then they're not geometric isomers. They're the same molecule. If they're trans and trans, the same thing. Okay? Let's try practicing with this. So let's draw a couple of structures here. So we'll draw two molecules. We'll put some additional groups on here. And we would normally do our first step to make sure that we have the same molecular formulas. I'm going to tell you the way I've drawn them up here. They are the same molecular formula. They have the same bonding pattern. So up here we have 3 bonds to hydrogen. As you go through and compare them, you'll see that all of them have the same bonding pattern. Even right here, it's really easy to identify the double bond pattern. That's why I like geometric isomers. So notice I have this carbon double bonded to that carbon. It has one hydrogen here, one hydrogen there. Same over here. They're the same bonding pattern. But now, which side that hydrogen on is on does become important. So as we start to look at these, here's my double bond going across there, and I find that my two hydrogens are both on the same side. That means my two carbons must be on the same side. And so this I would identify as a cis double bond. On this side, here's my double bond. I draw a line through the middle. One hydrogen is on one side. The other hydrogen is on the opposite side, which means the carbons must be on the opposite sides as well. This carbon would be pushed over here, this one pushed over there. And so this would be a trans double bond. So I look at that and say, okay, they were stereoisomers, and one is a cis double bond, the other one is a trans double bond. These two molecules then are geometric isomers. And that's all it takes. You look for the double bond, see if one's in cis and one's in trans. The double bonds do have to be in the same position, right? Otherwise, that violates our first rule of them being stereoisomers. But if they're in the same position and but opposite orientations is what we call it, then they're geometric isomers. And that's what a geometric isomer is. Okay? It contains double bond chiral centers in opposite orientations. Now, that was one type of chiral carbon. Let's talk about the second type of chiral carbon that we might see. And this is going to be the basis for our other two words then. So up here, we learned on this branch, we were looking at carbon-carbon double bonds. On these two branches, we are going to be looking at what we call asymmetric carbons. And that's the second type of chiral center that we see, or chiral carbon. So now we'll do number 2. Asymmetric carbons. I find that double bond carbons are usually fairly easy for Bio 180 students to grasp. Asymmetric carbons get a little bit harder. Okay? So by definition, an asymmetric carbon is a carbon bound to 4 different groups. Okay? And groups can be a single atom, like hydrogen or chlorine, or it can be a collection or a group of atoms together, like a hydroxyl group or a phosphate group. Okay? So to be an asymmetric carbon, the carbon, there's 2 points that we have to look at. It has to be bound, have 4 separate bonds, and each of those bonds have to lead to different groups. So let's come over and practice a little bit with this identification and see if we can identify some asymmetric carbons. So we'll just do 4 carbons up here. Okay. Now, the question we want to ask is, are any of the carbons in this molecule asymmetric carbons? Well, the way we answer that is we're just going to apply our definition. So we look at carbon number 1. And by the way, as you determine asymmetric carbons, you have to look at each carbon one by one. Each one has to be considered differently or separately. So we look at carbon number 1 right there. And we count the number of bonds, or the number of things it's bound to. So we've got a hydrogen. A lot of times I'll even draw, when I'm first starting out, I'll draw circles around it. So I've got 1 bond, it's bound to a hydrogen. Up here, I've got 2 bonds to an oxygen. And then this fourth bond right there, what's the group? It's everything that is attached to. And that's where beginning students most often commit a mistake. They look at this bond and they say, oh, it's bound to a carbon. It is bound to a carbon, that's true, but it's also bound to everything that that carbon is attached to. So this is actually a group, not that single carbon. When we look at this, we see, oh, they're all different, but there's only 3 of these. So that violates our first rule that it has to be bound to 4 different things. So that is not, we'll put a little X there, that is not an asymmetric carbon. Now we move on to carbon 2. So now I have to erase this line because carbon 2 is bound to different things. So we'll erase these groups. By the way, we can do a shortcut here. Anytime you see a double bond on a carbon, it's not asymmetric. Okay? That's the quick way of doing that. Now we look at carbon number 2. We're going to do the same thing. We're going to say, okay, it's bound to this hydrogen, so that's one group. Over here, we have another bond. We circle the whole thing, and everything gets attached to. That represents group 2. Right here, what it's bound to, that oxygen, and whatever it's attached to, this hydrogen as well, that's group. And then right here, that would be the 4th group. Do we have 4 groups? Yes, so we met our first rule. Do any of those groups look the same? And hopefully if you're looking at that and you've watched Sesame Street before, you can recognize that those are not the same combinations. So this one would be, let's put a check mark right there, that would be an asymmetric carbon. We'll erase these and we move on to the next carbon. If you look at that molecule for just a second, I'll do these in green. I'm not going to outline it because eventually you don't want to be drawing on your paper all the time. Eventually you'll get good enough that you'll say, okay, this is one group. This is another group, that whole thing. Here is a 3rd group. And that's a 4th group. And then I'll look at those and say, do any of those look the same? Now, right here, these two groups are different, but those 2 groups are both hydrogen. Those are the same. Therefore, this is not an asymmetric carbon. And then finally on the end here, if I do the same thing, here's one group, there's one group, 3, 4. I look at those 4 groups in my head. All of those are different, so that 4th carbon is an asymmetric carbon. Okay? Now, the ability then to identify asymmetric carbons becomes important when we're considering whether something is a diastereomer or an enantiomer. However, we're not done yet. We have to look over our 2 pairs, and let's practice this ability to identify asymmetric carbons with the pair of molecules that we're trying to differentiate and apply it. We already know, if we look over them, there's no double bonds, so we're not even considering geometric isomers in this case, because there's no double bonds. So I know it's not geometric isomers. To be enantiomers or diastereomers, I have to have asymmetric carbons. They're the requirement. So I'm going to look at these molecules and see if I find any asymmetric carbons. And we're going to do like we did up here. We're going to put an X or a check if they're asymmetric. So I look at this carbon, double bond, so that's automatically an X. It's not asymmetric. Here I've got an OH, an H, that group, and then this whole rest of the molecule. That one is asymmetric. Right here I've got 1 group, 2 group, 3 group, 4 groups. That one's also asymmetric. Right here, immediately I look, oh, 2 hydrogens. Those are the same. That one's not. I don't even look at the other groups because I've got 2 that are already the same. Right here, I look, I've got an OH, I've got this big thing right here. Oh, there's 2 hydrogens right there, so those are asymmetric, or not asymmetric as well. Over here, because I already established, remember in step 2 I said this has the same bond pattern? So I don't really have to look. You can do it if you want to, but I already said they're the same bonding pattern. Therefore, if they were asymmetric over here, they're asymmetric on that pair as well. Now, here's what we have to look at. Asymmetric carbons can be in opposite. We often refer to this as mirrored, and I'll show you why in a second, or same orientations. Remember before with geometric isomers, we said that it could be in a cis orientation, or we said it could be in a trans orientation. Later on, when you take the organic chemistry class, you will learn that asymmetric carbons can be in what's called an R orientation, or they can be in an S orientation. We're actually not going to get into how you differentiate R and S in this class, but we will be able to tell whether it's in the same orientation or the mirrored orientation, and I'll show you how to do that on the board in just a second. But for a minute, let's talk about what we mean by the same or the mirrored orientation. So here I have 2 models right here, and it has a central carbon bonded to 4 different things. So we've got a red atom, a yellow atom. In fact, I'll remove this hydrogen. We've got a red atom, a purple atom, a green atom, and a yellow atom attached to these. Now, what we mean by you could either be the same orientation or a mirrored orientation. These 2 have the same bond pattern on them, the same rotation around that carbon. What that means is they're superimposable. You can see I can orient these 2 so that if I could merge them, they would exactly overlap each other or be superimposable. Okay? That would be what we would call the same orientation. However, if I break and swap 2 of these bonds, I still have the same molecule. Everything's still bonded to each other, but now they're no longer superimposable. If I try and superimpose the red and the purple atoms, notice that my yellow and green are now opposite each other. They're going to clash with each other. Okay? And that's what we call a mirrored orientation. When it's mirrored, I can't superimpose them, but I can orient them in such a way that it would look like there was a mirror along that plane. So now you could see, hopefully right here, that this would be the mirror. So these 2 would be in a mirrored orientation to each other. One would be R and the other would be S. Okay? And so that would be a mirrored pair, is what we call that. All right? Let's go through now and look at our 2 molecules. And what we want to do is we're going to use what we've learned about asymmetric carbons and being opposite or mirrored orientations. And we're going to look at our 2 carbons, and we're going to compare them and see if they're in a mirrored orientation or if they're in the same orientation. Now, one trick to this, as we go through and compare them, if it's not asymmetric, we don't care. If it's not asymmetric, there's no mirrored or same. There's a way, if they're not asymmetric, there's a way of rotating them so that they would be the same. Okay? So up here, we said those weren't asymmetric, so we don't even care what those are. I'm just going to put a dash there. No one cares. These 2 are asymmetric. So are they in the same orientation or a mirrored orientation? Well, I have the OHs on each side. They're on the left side of the carbon, and the H's are on the right side, and then the tops are the same and the bottoms are the same. So I'm going to put an S there because those 2 are in the same orientation. Does that make sense? Do you guys think you could recognize that if we had a different problem? Now, what does a mirrored look like? Well, mirrored would look like this. Notice I have the nitrogen on this side. The nitrogen, here it's on the right. Here it's on the left of that carbon. This would be a mirrored orientation. If I had a mirror right here, then the nitrogen would be mirrored, and then the hydrogens far away would be mirrored on each other. So that would be a mirrored orientation. This one, not asymmetric, no one cares. This one, not asymmetric, no one cares. Now we can identify the relationship between these two molecules using the following key. If, right here we have all pairs are mirrored, then we have enantiomers. The definition of enantiomer is having a mirrored orientation at each asymmetric carbon. So if we got through drawing our picture and we had M, those two we would know would be enantiomers. They all had the same orientation. Diastereomers, di means two, or a mixture. And so diastereomers occur when we see a mixture of mirrored and same orientation. And it doesn't matter how many S's or M's there are, as long as there's at least one S and at least one M, then it's a diastereomer. Now, one other possible combination can occur from this, right? So we could have all mirrored. We could have a mixture of mirrored and the same. The final combination is we could have all of them be the same orientation. What if we had SSS up there? Well, if they're all the same, then those two are actually identical molecules. It's actually the same molecule. Okay? It might be rotated at some non-asymmetric points, so it might not look exactly the same, but there's a way of twisting it to be the same. Okay? If we look at our combination that we have up here on the board, we find that we had one S asymmetric pair and one M asymmetric pair. Hopefully, using our definition tree over there, our decision tree, you would say, oh, these then must be diastereomers. And those two molecules are, in fact, diastereomers. Okay? So, let's summarize for a second what we've learned, and then we'll put some different molecules from the worksheet on the board, and I'll walk through one or two examples of going through these steps. Okay? So, we have learned that there are different types of isomers. So we have just isomers, and then isomers can be divided into stereoisomers and structural isomers. And then stereoisomers can further be divided into geometric isomers, diastereomers, or enantiomers. And then we've learned several questions that we can ask to determine what the relationship is. And so we just go through that process of steps, and we analyze the molecules in front of us. We identify the asymmetric carbons. We identify double bonds if there are any, and compare and contrast them to come up with an answer to the question of what is the relationship between that pair. Okay? What we'll do is one final wrap up. We've been talking about different questions that we would ask. And so let's give that process to you. So when you're faced with determining a pair of molecules and what the relationship is, the first question we're going to ask ourselves is, do they have the same molecular formula? That's the first question. A lot of students will want to skip this question. So they'll look at the pair of molecules, they'll see a double bond in it, they'll, oh, I know, that's a geometric isomer, and they'll jump right to geometric isomer, when in fact they didn't even have the same number of carbon molecules. They just happen to have two double bonds. Okay? Don't forget this first step. You have to do the process in order. If you skip one of the steps, then there's a good chance that you may get it wrong. Your teachers will be able to trick you. If you do each step, you'll never get it wrong. So first step, same molecular formula. Count carbons. So we're going to count carbons, then oxygens, nitrogens, hydrogens, and then whatever X's, whatever elements there might be. The second, we ask ourselves, do they have the same bond pattern? Okay? That was, remember, the same molecule, same atoms or elements are attached to each carbon. Okay? Do they have the same bond pattern? The third question we asked, and we only asked it if they had the same bond pattern, was we asked if there were any chiral centers. Do we have double bonds is usually the first one I look for. And the next one would be asymmetric carbons. In question 4, this applies only to really double bonds. I asked if they were opposite orientations. Meaning, were they cis and trans? And then if they were, then I'd have geometric isomers, and I could stop there. Question 5 really applies to asymmetric carbons, and actually I'm doing the same thing. I am asking whether the asymmetric carbons are in opposite orientations. And if they're in opposite orientations, R and S, again, we haven't given you that, but you can look at them and tell whether they're mirrored from each other or whether they're the same. And that's how you determine the opposite orientation. And then based on the combination of opposite or same orientations, mirrored or same orientations, then you can determine which type, whether it's a diastereomer, an enantiomer, or the same. So we're going to then just apply this on some practice problems. Okay, so now that we've learned some of the principles associated with isomers and their identification, let's go ahead. I pulled up on the board a couple of sample problems from the worksheet that you have available to you. So these are the first 2 sample problems. The question is the same. What is the relationship? Here's a pair of molecules. What is their relationship? And the answers will all be over here. They'll either be non-isomers, they will be structural isomers, or some form of stereoisomer, or they could be identical molecules. So for each pair, take a second and go through and determine what you think the relationship is between that pair. Now, in this example, I have made little gray boxes here to kind of help you with the mirrored or the same pairs and the asymmetric carbons on identifying those. You usually wouldn't see those gray boxes there. We just added those to help you go through the exercise. Okay? So take a second and go through and do that. Let's look at this first pair. And the first thing we want to do is count the atoms and look at the molecular formula, see if it's the same. So I start with carbons. I have 1, 2, 3, 4 carbons on that one. So I can write down here C4. 1, 2, 3, 4 carbons there. Looking good. I come down to the outside. I have, let's go with oxygens next. So I have 1, 2, 3, 4, 5 oxygens. O5. 1, 2, 3, 4, 5 oxygens on that side. Next, I'm looking through. I don't see any nitrogens. I am noting that there's a couple of chlorines there. So let's go ahead and count those. So chlorine, I've got 1 chlorine, 2 chlorines. So Cl2. Over here I go through, I've got 1 chlorine right there, and I've got a second chlorine. So that's Cl2. No differences so far. The only other atom I can identify is hydrogen, so let's count those. 1, 2, 3, 4, 5, 6, 7, 8. So H8. 1, 2, 3, 4, 5, 6, 7, 8. H8. So those two molecules have the same molecular formula, and I've done number 1. They must be isomers. Next question. Are they structural isomers or are they stereoisomers? Well, I look at this carbon. It's got a Cl, an OH, an H, and then the rest of the molecule. This one also. So those two seem to be in the same pattern. OHCH, OHCH. I already determined that those two were the same. So that looks good. Here I've got OH, 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 OH. These two are already determined to be the same. And my fourth carbon, I've got an OH and a Cl. I've got an OH and a Cl, hydrogen at the bottom. These both have the same bonding arrangements. Every carbon is attached to the same type of molecule. It may not be the same rotation around the carbon, but it's attached the same type of elements on every carbon. So that's step 2. Now I'm looking for chiral centers. Remember, double bonds are easy. There's no double bonds. I can skip that. Asymmetric carbons then becomes my next question. Asymmetric carbons. So right here, here's my first carbon. Here's 1 group, 2 groups, 3 groups, and then this big thing is the 4th group. All 4 of those are different. That one's asymmetric. Next carbon, I've got an OH as 1 group. That's different from H, which is different from that, which is different from that. Asymmetric. This one, I've got an OH, I've got an OH. Those are the same. And right here, I've got OH, Cl, H, and then a big group up here. Those are different. Since I already determined that they had the same bond pattern in the previous step, I don't have to redo everything. I can just go ahead and check all those boxes, the same pattern as before. Now, all I have to do, I've identified my chiral centers. Now all I have to do is look at the orientation of those chiral centers. So up here, I've got an OH here on the right. My OH is there on the right also. Those are the same sides for that particular group. So that has the same orientation between that asymmetric pair. Right here, I've got an OH on the left, OH on the left, also the same orientation. Here, I don't care because it wasn't asymmetric. And on this last one, I have got chlorine on the right, and here the chlorine is on the left. So that's actually a mirrored pair. I go through and I look at that. I use my formula. I've got 2 S's and 1 M. That's a mixture. Therefore, this must be a pair of diastereomers. And that's how you go through and apply the rules to identify a pair of molecules. You see, as you start doing it over and over, the steps at first seem difficult, but you can actually do these really quickly over time. Okay? The longest part sometimes is just depending on how big the molecule is, counting the carbons and the hydrogens, the oxygens.